Hello, welcome to part 3 of chapter 15, where we're going to talk real quick about life cycles. So, a life cycle is a sequence of events that produces the next generation. And if you're a sexually reproducing organism like humans are, this more often than not is going to involve the alternation between haploid and diploid organisms, or between a single set of chromosomes and the double, triple, quadruple, or whatever number set of chromosomes a eukaryotic organism has. So if a species is considered diploid dominant, which most animal species are, then their haploid cells are going to be a specialized type, and we refer to these as the germline or germ cells. A haploid dominant species, which are a lot of fungus, some protozoans are also haploid dominant. And there are a few insects actually, where the multicellular organism is actually in a haploid state. And haploid cells will form a diploid zygote and then immediately enter into meiosis to create haploid cells. And with fungus, that usually are the haploid spores. So a lot of it will depend on how long or how much of the life cycle the organism actually spends in a haploid versus a diploid state. So this is referred to sometimes as what's known as the alternation of generations. Plants and algae do this a lot, and this is due to what's known as intermediate dominance. In fungi and some plants, if it's a multicellular organism that is diploid, the germ cells are the sporophytes, or it is classified as a sporophyte. But if the multicellular form of the organism is haploid, then it's known as a gametophyte. And you'll see this mostly in fungus. Now, you will study alternation of generations in your next biology course, but the major difference between a sporophyte and a gametophyte is whether one is diploid or one is haploid. Now, among different species, relative size of the sporophyte and gametophyte, how big they are, what part of the life cycle they are, will be variable. So here we can see a couple of different life cycles, right? We have our haploid dominant, diploid dominant, and then alternation of generations. And in blue is the haploid state, and in purple for this organism, right? Purple is haploid and blue is diploid. So you can see that this is a haploid dominant life cycle because more than 3/4 of the life cycle is in a haploid state. Over here in a eukaryotic, like we are, you can see that 3/4 of the life cycle or more is in a diploid state. So we exist in a diploid state, so we are considered diploid dominant. Alternation of generations, it is 50-50. So the life cycle, they spend half their time in a diploid state and half their time in a haploid state. So you can see in the haploid state, they are in a gametophyte. And in the diploid state, they're in a sporophyte. So this is a fern, for example. Ferns are ancient plants, very old plants. And when we see a fern, like they call this a fiddlehead, and all of these different parts, the leaves, when we see the actual plant stage of it, it's in a diploid state. It's a sporophyte. But on the back of fern plants, on the back of the leaves, are these little brown dots called sori. And in these sori are spores. The spores are haploid. And the spores will spread by wind, and they'll form this gametophyte form of the plant, which then fertilizes, creates sperm and egg, fertilizes, comes together, and creates the diploid plant that we see growing in the forest. Now, other anomalies that occur are variations in chromosome numbers. So how many chromosomes a given species might have it may be different from one species to the next or one genus to the next. However, it will be constant within the actual species itself. For example, humans have 2 sets of 23 chromosomes for a total of 46. This is the normal number of chromosomes for a human being. This does not really vary much outside of chromosomal disorders. This number doesn't vary from one human to the next. Dogs have 78 chromosomes. They have 39 sets. Fruit flies have 8 chromosomes, or 4 sets, and tomatoes have 12 sets, or 24 chromosomes. So you can see dogs have a whole lot of chromosomes, have many more chromosomes than we do. Chromosomes are identified by their size. So we have what's called location of centromere, and a banding pattern. Now, we looked real quick when we, in one of the previous lectures, we looked at the creation of a karyotype. And when those chromosomes are spread out on a slide and stained, we're going to see some physical differences. And this is how they're matched up. Now, in terminology here, location of a centromere. Centromere, remember, chromosomes kind of look like this when they're condensed. And they have this kind of knot in the middle. We call this the centromere. It's kind of like where the chromosome pinches. Like that. So it's called a centromere. Now, some chromosomes have a really long edge and a short edge. Others will be right in the middle. It all just depends on where the centromere is. So we first look at the size of the chromosome. We identify them by size. We then look at where the actual centromere is located. Now, if I have a chromosome that's acentric, so I have a long arm, this is known as the long arm, and this is the short arm. The short arm is called the P arm, and the long arm is called the Q arm. When we line chromosomes up, we line them up with our P arm on top. So this is the P and this is the Q. If the centromere is smack in the middle, like we have over here, then it's called metacentric. If it's off-centered, it's called submetacentric. If it's really close to the end, it's called acrocentric. And if it's at the very end, it's called telocentric. Look at some pictures of it. Also, when these chromosomes are spread out on a glass slide, they are stained. They use a stain called Giemsa stain, and this provides a characteristic known as G banding. Okay, so here we are looking at the different shapes. This is just an illustration of what I was saying in the previous slide, right? We're looking at the location of where these centromeres are. And of course, we have the Q arm is long and the P arm is short. Think of P for petite. Someone is petite, that usually means they're very small. So the small arm would be the petite arm. When we that's in comparison of the locations. This is what's known as Giemsa staining. They use this reddish type stain, and where chromosomes are really, really super condensed, the Giemsa stain really concentrates in that area, and so it gets much darker, and so we can see these really nice dark lines. So they compare where the Giemsa lines, this is called banding, and so they compare where these G bands, Giemsa bands are, as well as the centromere in order to locate the two of them together. So they're just showing you an illustration right here of what that set of chromosomes looked like really close up. Remember, one is from mom and one is from dad. This, by the way, is a female because we have two X chromosomes. Now that brings us to the idea of chromosomal mutations, and we do have mutations, of course, at the genetic level, right? We have DNA nucleotides can be replaced or changed. Different types of mutations can occur in the DNA, but we can also have entire chromosomal mutations as well. These chromosomal mutations can result in deficiencies. This would be where we have a deletion. So you can see here we have a region, they're just showing you this region here. If the chromosome has disappeared, it was cut out or is missing, something happened to it during replication, it's no longer there. So the resulting chromosome is much shorter. This is a deficiency. The genes are missing. Now these deficiencies, remember, can sometimes be overcome or not necessarily noticed because you have one from mom and one from dad. So if the one from mom is deficient in a region, the one from dad will still have that region, so you still will express whatever genes are on that second chromosome. That's why we have pairs. The issue can be either you get diminished function because you're not producing enough. The other could be that if there's a mutation in dads, then you're only inheriting one copy of the genetic mutation. So you have a combination of a chromosomal mutation and a DNA mutation or a gene mutation. In duplications, a section will occur 2 or more times in a row. So in this case, it's the exact opposite. We're actually including an extra piece. These are all issues from crossing over. If you think about that process of crossing over that we talked about, that's where we can end up with some deletions, or a lot of times this duplication where the crossover event occurred, but instead of them actually swapping regions, this guy actually just got his region, and so this is a deletion and this is an insertion. Then we have what's known as an inversion. And an inversion, here our chromosome, let's say, was in this direction, and during the crossover event, for some reason it gets flipped, and now the genes are down here. So if I had a gene on this chromosome right here where this white band is, instead of it being located here, it's now located up here. And that can make a difference in whether or not my transcription factors, my replication machinery, and transcription factors such as silencers or activators are going to be able to transcribe or translate these genes. So it can alter the phenotype of this particular chromosome. And then we have translocations. And in translocations, we have a simple translocation and a reciprocal translocation. In a simple translocation, we have some form of the chromosome. We move one segment of a chromosome to a different chromosome. So we go from chromosome 1 to chromosome 3. Right? So this can be within the same chromosome or in a different. In a reciprocal, we get an exchange between 2 different chromosomes. So what happened here is our chromosome moved over and attached to another chromosome, but this guy remained intact. In our reciprocal, it reciprocates the exchange, and so now I have them basically 2 different chromosomes, chromosome 6 and chromosome 10, or chromosome 1 and chromosome 5. I have 2 completely different chromosomes exchanging genetic material, exchanging pieces of their chromosome. Whereas in a simple translocation, I just have one donating to another. We can also see changes in chromosome number. Euploid is a chromosome number that's viewed as normal. So for us, we are 2n, right? So we are 2 times whatever number of chromosomes we have, which is 23. So that's a total of 46. So we have pairs. A polyploid is 3 or more. So if you're diploid, you're 2n. If you're triploid, you're 3n. And if you're tetraploid, you're 4n. Strawberries, for example, are octoploidy. So they are 8n. They have 8 copies of each of their chromosomes. This is why we use them as a sample DNA extraction material. When you're learning to extract DNA, we use strawberries to initially learn the process because they have so much DNA. It's much easier to extract. Aneuploidy is where there's an alteration in the number of chromosomes. So they don't really have, they go by a total number of chromosomes. They don't really have an exact set. Trisomy is when we have a normal 2 copies of a chromosome, but then we end up with a third. So trisomy is called a 2n plus 1. This happens in Down syndrome. We have certain sets of chromosomes where we're normally 2n for all of our chromosomes except for 1, and then this one set will be 2n plus 1. So we end up with an extra chromosome, like an extra chromosome 13 or 16 or 18. So we end up with an extra copy of a single chromosome. So our normal 46 becomes 47. Monosomy is when we are missing one. So this would be 2n minus 1. Then we have the property of nondisjunction. And in nondisjunction, aneuploidy over here, or trisomy, monosomy, and aneuploidy is the result of nondisjunction. In nondisjunction, we get improper sorting of our chromosomes. So you can see here in normal meiosis, each of the haploid cells from this diploid cell would get 1 red, 1 green, and 1 purple. So that's what we have here, and these are our normal gametes. But in nondisjunction, the red ones here, they didn't separate properly, either at the centromere or during some form of cell division. So this gamete is missing. So any of the offspring from this one would be monoploid, right? Would be mono. And this one has an extra. So any of the offspring from this gamete would be trisomy. Then we have the property of interspecies breeding. And with interspecies breeding, organisms that are allouploid have at least one set of chromosomes from two or more different species. An example of this might be they were trying to reintroduce the Florida panther. So they had to find another cat species, another wild cat species that was as closely related and had the same number of chromosomes as the Florida panther, which they actually found to be the Rocky Mountain mountain lions. And so they were able to interbreed mountain lions with Florida panthers to try and keep the Florida panther gene pool running throughout Florida, try and bring the Florida panthers back. So an allouploid organism has at least one set of chromosomes from two or more different species. So you could say ligers, right? Tigers and lions. We also have mules. Mules are a cross between a donkey and a horse. Allodiploid is where only one set of chromosomes are from two different species. Allopolyploidy is two or more complete sets of chromosomes from two or more different species. And allotetraploid is two complete sets of chromosomes from two species for a total of 4 sets. Now, this will work just fine for most organisms as long as they have the same number of sets of chromosomes. So here we have an example of alloploidy, allodiploid, and allotetraploid. So an alloploid, right, we get one set from one species and one set from a second species to give us 2N. If you notice, however, these don't match because they're different species. So this one might match with this one, but these two don't necessarily match with these two, and we end up with an extra chromosome. In allopolyploidy, we get both sets. So instead of going from 2N to 1N, we go 2N to 2N. Same thing here, 2N to 2N. And so we get a massive number of chromosomes here. So there are some consequences with this allodiploidy and alloploidy, and that's because we as animals, eukaryotic organisms, don't do very well. We don't usually tolerate deviations from being diploid, from being 2N. There are some exceptions, but most of the time it's what's called a lethal consequence, meaning it does not result in viable offspring, or the offspring will not survive for a long period of time. There are a few deviations, though, that are important. One is in honeybees. For honeybees, male bees only have one set of chromosomes, and female bees are diploid. So a male bee will be haploid, female bees are diploid. Now, haploid bees, the male bees, their only job is to leave the hive, go to a what's known as a drone center, which is an area in which drones will congregate. The drones are much larger, and it's usually 30 or 40 feet up in the air. It's usually above a feeding area, and the drone bees will congregate in this area in order to meet with queen bees. So it's kind of, you could say it's kind of a meeting place. It's kind of a little club for drones and queen bees for mating to occur. A queen bee will mate with multiple drones in a 24-hour period, and all of the sperm from all of those different drones will be retained within the queen bee for the rest of her life. So she will mate that one time, or for that one period of time, and then she will go back to her hive, and she will preserve those sperm in her abdomen in order to fertilize all of those eggs for the rest of her life. So it's the only time she ever leaves the hive is to go mate with drones. Drones, once they have mated, die. They do not return to the hive after mating. So the only purpose for a drone bee is reproduction. So they don't have multiple copies of chromosomes. They just simply have a single set. There are some diploid and polyploid species of different amphibians and a few reptiles as well. Plants often exhibit polyploidy. Ferns, a lot of flowering plants, wheat, all of these polyploidy in plants is usually very well tolerated, but in animals it usually is not, with a few exceptions. Aneuploidy in all eukaryotic organisms is known to be detrimental. Trisomy and monosomy have an imbalance, and this is because gene expression, we have, remember, with, let's say, trisomy. I normally have 2 sets of all my chromosomes, but then all of a sudden in one set I have 3. Now, what's known as dosage compensation, which is the idea of, there we go, the idea that this allele will balance this allele in gene expression, gets thrown off when we have trisomy. So when I have 3 copies of the chromosomes no longer balance each other. That 3rd chromosome is really changing the expression of those genes that are located within that chromosome. So it often interferes with cell function, cell production, development of the organism over time. An example of trisomy is Down syndrome. So about 5 to 10% of all eggs that are fertilized in humans result in an embryo that has some kind of chromosomal abnormality, usually the number of chromosomes. Nondisjunction is not all that uncommon. About 50% of what are known as spontaneous abortions are due to alterations in chromosome number. These are usually occur without the mother ever even knowing that she was pregnant. So it occurs upon fertilization. There are some abnormalities that can be survived, such as trisomies or abnormalities in the number of sex chromosomes. So here are some examples of aneuploidy that is tolerable in humans. Trisomy 21, 18, and 13. Trisomy 21 is known as Down syndrome. Trisomy 18, meaning we have 3 copies of chromosome 18, is Edwards syndrome, and trisomy 13 is Patau's syndrome. All 3 of them have very similar consequences. Then we have aneuploidy in the number of sex chromosomes. One thing to notice is you can survive without a Y chromosome, but you cannot survive without an X chromosome. All of these have X chromosomes, whether it's XXY, X2Ys, triple X. All of these require the X. If you have a Y0, this is lethal, and the fertilization won't even complete. So understanding aneuploidy is important. Oh, sorry about that. Understanding aneuploidy is important in terms of nondisjunction and chromosomal disorders. Hopefully this finishes up this chapter, chapter 15, and I will see you in the next one. Have a good day.